Okay, hi everyone. So we're here for our first in-person meeting in a while indoors. Thanks all for attending. Michelle Slayton, our usual person who would be hosting our vice president, couldn't make it today, so I'll be just subbing in for today's talk. And so I get the pleasure of introducing our speaker, Robert Klinger. A little bit about him. He is a avid tennis player, skier / rabid Dodgy Dodgers fan.
Oh yeah, can we get game day on MLB Network so I can
But on his spare time or real time, he works at USGS as the ecologist and current incarnation as a statistician, though he likes to call himself a very good quantitative ecologist, but USGS begs to differ. So
yeah, they think I'm a terrible
so Rob's been a world traveler, has studied a lot from brief stints in the Galapagos to longer stints in tropical forests of Belize. And in the last 5 years, he's set down roots and he's studied plant communities
sounds off.
fire effects in the Mojave Desert.
Well,
up to plant communities with small mammal interactions and climate effects in the Sierra Nevada. So that's what he's going to be talking about today. So without further ado, I'll leave it to Rob.
Plenty of ado, thank you. Thanks for coming out live and in person. Put on my spectacles here. The first thing that I want to do, this is, if you count the year of the pilot study, this has been ongoing now for about 14 years, almost 15 years now, this study. And there have been waves of people that have helped out on this. This certainly has not just been a Robert Klinger project by any stretch of the imagination. Tom Stevenson, who I think some of you probably know, Tom is in charge of the Sierra Nevada Bighorn Sheep Recovery Project, and I think deserves a medal for what he's done for that program. He and I have had a real ongoing interest in the meadows for a variety of reasons. But then there have been all these other people that have been coming through, and the other three that I'm listing here, Sarah, Logan, and James, are these three young brainiacs that have really been moving things along, helping me with satellite data over the last few years. I want to give a huge nod of appreciation. If this thing goes forward, come on, advance. There we go. I had an incredibly good crew. I had a USGS office here up until the sequestration in 2013, 2014, had to shut it down, but great field biologists that did some tremendous work collecting the bulk of the field data on this, and then support from a number of people in different agencies and universities throughout this time, if nothing else, kind of like a brain trust. And the bulk of the funding came from the USGS National Climate Change and Wildlife Science Center. So this certainly has been a big effort. So unless you've totally gone down the faux news, one IQ point Newsmax rabbit hole, climate has been changing. It's going to continue to change, and we have to learn to deal with it. Nevertheless, where I'm going with this is just because climate is shifting, it doesn't mean that ecological processes just stop. They might get modified, they will interact with climate, but they're going to continue on. And I'm going to be focused on two basic ecological concepts: variability and resistance. And we're going to have a little audience, just a little audience participation to kind of solidify what I mean by those. And I'm going to be looking at the meadows in different ways and at different scales using A lot of data that we collected from plots on the ground and a lot of data that we've pulled in from outer space, from satellites. So depending on where you draw your lines, about 3 to 10, 11% of the upper elevation zone of the area is comprised of meadows. Okay, so not a very large area, but they are disproportionate in their importance, and that's in many ways. People think of them commonly in terms of water storage, runoff, and things like that, hydrology basically, but they're actually the centers of plant diversity in the high elevation zone and really valuable habitat and resources for both vertebrates and invertebrates. So very, 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 very valuable, significant ecologically. And not just in the Sierra, but globally, because they're high elevation. There's a lot of concern about them, in regards to their vulnerability and that they're going to change. Two of the words that kind of get me. So let's think a little bit about vulnerability and change. There's nothing wrong with those terms inherently, but they come from kind of a value-based conservation perspective. They're very vague. So what is it exactly that people mean when they talk about being vulnerable or things are going to change? Are they talking about the species composition in the meadows? Are they talking about the diversity? Are they doing the functional attributes? Maybe the structure of them, they're going to change along the meadows. It's all very vague. So being annoyed at this, I stepped back a little bit. I said, well, let's just look at this objectively from an ecological standpoint. And what people, I think, are really trying to get at is this notion of stability. Now, ecologists, just kind of two major groups of concepts regarding stability and how it's defined. A very commonly accepted one is this multifaceted notion that is made up of these 4 components. So you've got resistance, resilience, persistence, and again, variability. But these aren't mutually exclusive. They play into each other. If you've got a lot of resistance to change, you're going to persist. If you're really variable, there's a certain amount of resilience in there. And you go back to some kind of dynamic equilibrium. Sometimes, not always. So when people are talking about change in these high elevation meadows, generally they're thinking of two pathways. One basically comes down to higher rates of evapotranspiration. Things are going to get drier. And that's going to change the functional attributes of the meadow in terms of less biomass being produced, or it's being produced at lower rates, or it's drying out at higher rates. Or there's these big shifts in species composition. Following from that is the second major pathway, as because of this drying effect, this is going to make them more susceptible to colonization from woody species moving up in elevation. So that notion is kind of a deterministic one, the climate is king view of the world. So here's an elevation gradient with our pine forests down in the subalpine lower elevations. And then our meadows kind of assemble along the moisture gradient in the upper elevations. This is kind of the standard viewpoint of things. And temperatures are rising and evapotranspiration is increasing. And so that creates better conditions for the conifers, and they move up in elevation, colonize the meadows, and then there's these transitions. Now, this could play out, very well could play out. But I think that if you just think of how ecological systems operate, a broader perspective might be a little bit more insightful. That there are multiple factors that are interacting here. So things such as landscape characters, topography, soil, biological interactions like competition, facilitation, and another one that I'm going to get to in a moment are really important here. And these are often overlooked. So I called this, the world is more complex than a single factor perspective. And you can flip things around easily. And there's actually evidence of this from other parts of the world, that you can get increased productivity in these meadows because you have a lot higher rates of photosynthesis, not growing seasons. And what that might end up meaning is increased competition.
Listening to the CNPS talk that I couldn't go to.
In these meadows. And this is the one that I think people just in the US, it's more appreciated in Europe and Asia. But at least in North America, we haven't really appreciated this as much as we should. And that's that animals and mammals in particular are really important when it comes to herbivory. They eat things that are small, like seeds and seedlings. And there is elevation or evidence of this from both high elevation, high latitude systems in other parts of the world. So it raises the questions of could the mammals potentially decouple this climate-driven transition of meadows to more woody patches. All right, now here's the small audience participation.
Listen to the woodpeckers.
Page or the same trail as me with all this variability and signal and noise stuff. All right, let's say that about 40 years ago, your grandparents went up to the high Sierra, high parts of the Sierra, and they started to measure something. And let's say they did that for about 10 years, and this is what they found. First 10 years. All right, so what's going on here? Audience participation. What do you see?
What's the label on the vertical?
It's abstract. That could be any kind of anything that you measure. It could be a population, it could be a rate of something, whatever. Would you say there's a trend here? An increasing trend? Yeah. All right. So now let's say that your parents picked up where your grandparents left off. And then the next set of years, and if you're really sharp, keep an eye on that y-axis and the units on the y-axis. What's going on here? Decreasing. Yeah. Going down a little bit. Good. I like that little bit. Okay, now you come along and you do it for about 6 years. And this is what you found. What's happening here? Kind of, maybe, sort of. Something's happening there. There's your kids. What do they find? Yeah, yeah, almost went to zero. And then there's your grandkids. So the point here is, with these 5 segments, how you interpret this change over time depends on where you're standing, so to speak, at any point in that timeline. That's what it looks like over the 40 years. Now, what an ecologist might do, actually most ecologists would do something like this. They'd fit some kind of curve through that. Do you see a trend there? I don't. I see dynamics. I see a lot of dynamics. Is this a useful way of looking at this? I don't think it hurts. It's not a bad thing. I would probably do something like this as kind of a first cut. Yeah. But what I'm going for, where I'm taking us in hopefully less than an hour, is a little bit more comprehensive look. That's the same series of points over that period of time. And now we're going to take a stability-based perspective on this. Okay, there's a period of high resistance. There's fluctuations, but it's really just kind of fluctuating around an average, what we call a dynamic equilibrium. But then, like you noticed, it went down almost to zero, almost quit persisting. But it bounced back up, and that's resilience. There's your variability. Different segments of that, and it's still here. So it has persisted. So keep this in mind as we work through these different aspects of the meadows. Okay, how about spatially? That was over time. Let's say your cousin did a 25-year study, and they did it at 50 different sites. This is what they found. What's happening here? Yeah, something's going down with time. Is that a trend? I think it's a trend. Now, let's say you broke that up, partitioned that out to your 50 sites or 50 populations, whatever that was. There you go. Still think there's a trend? I do. I agree with Marty. I think there's a trend. What about now? Yeah. This is extremely common. And what you see published a lot is the red line. You don't see everything going on around the red. There's not. And I think that is limiting our inference on how things are shifting in this crazy world of ours. Okay, now we're going to start moving to what we've actually been doing for the last 10, 12 years or so. The meadows are part of a larger project with a lot of moving parts. So we've been collecting all sorts of different data from outer space, satellite data, and we collected a whole bunch of data on the mammals and the plants, the meadow communities. Well, more than the meadow communities too. And then we did these experiments with the mammals, and all these data are feeding into different types of models, ultimately going to where we want to get to the point where we can make some reasonably informed projections, forecasts of where the meadows might be going over the next 50, maybe 100 years or so. So the meadows fit into this as a part, but a very important part. So all those purple amoeba-like blobs in the map, the green lines are our study area. And those amoeba-like blobs are the areas where we've been sampling the mammals and the meadows. So that spans about 3 degrees of latitude, a little over 3,500 feet in elevation. So that's a pretty good chunk of the upper elevation zone of the Sierra. The plot data are looking at things like composition and diversity in the plots. We're going to get into that in mere moments. The satellite data has been used mainly to look at dynamics and biomass, how much biomass there is, how fast it's being accumulated. And then the experiment is looking at these potential changes turning these meadows into woody dominated communities. Now, for those of you who have not been here for the last 10 years, just a little bit on background environmental conditions. It's also an opportunity for me to give a big shout out for this, who would you call him? I call him a geographical physicist. His name is Carl Ritter. And Carl started off as a grad student at UCSB. He went to Caltech, and now he's at the University of Colorado in Boulder. Carl has been doing for years now is pulling together data from different satellites, and he and his collaborators have come up with this beautiful layer that is daily estimates of the fractional snow cover in these 30-meter pixels throughout most of the West now. I think they've got it going back to 2000, right up to now. It's an awesome data set. So what I did is I said, okay, let's take a 20% threshold. Anything below 20%, we're going to call snow-free. And I calculate the number of snow-free days. And I could do it on a weekly basis, on a monthly basis, on a yearly basis. This is the yearly estimates. So that left panel is the number of snow-free days. And it clearly shows that, yes, we had a drought and we also had some big snow years too. So right on, Carl, your layers are working great. That right panel is data from the plots on soil moisture. And that corroborates, that lines up really nicely with the snow layers, and says, yeah, we had less soil moisture during the drought. You would expect that. So there's a signal, and of course that signal is real consistent, isn't it? Not necessarily. That left panel is showing the variation in soil moisture at the plot scale. That dashed line at 0 is the average, and then the little gray dots with the credible intervals around them. Those are your estimates of how close they were to the mean. And out of the 20 plots that these data came from, 4 or 5 kind of were in line with the mean, and everything was either drier or wetter than you would expect over that period of time. Same thing at the quadrant scales. There's this huge amount of spatial variability in soil moisture, even though we got a nice clear signal out of it. So there's a little foreshadowing for you. A little information on the vegetation data and what we collected and how we did it. All in all, we have 380 vegetation plots, and 180 of those are in meadows. 160 of those were sampled one time between 2010 and 2014. The other 20 were sampled 7 times over a 9-year period. We missed 2014, 2016. We're collecting all sorts of data in these plots, or we have collected all sorts of data in these plots. Most of the focus is going to be on the quadrat, these 1 square meter quadrats where we've got the cover and height, maximum height for the species in those quadrats. But I'm also going to pull in the biomass data a little bit from much smaller quadrats, and then just a little bit of the data on density of the conifers. So we had this extensive, in terms of the area we covered, and intensive sampling effort on the vegetation. Okay, so let's dive into composition. By that, I mean the species that are there and how abundant they are, what their cover was. Okay, I'm going to start off with the plots that we sampled once. I'm going to address 3 questions. What are the main influences on species composition, and how do these change with scale? How important of an influence is climate on the composition, plant composition in the meadows? And then, is there a spatial trend, latitudinal trend, longitudinal trend, elevation trend with this predictable turnover in species? All right, these kinds of analyses, they are not the most intuitive. You get kind of these blobs. So let me explain to you what's going on here. This is at the quadrat scale. And what I'm doing is putting in a suite of variables and seeing how well they predict turnover in species composition. And overall, this was a really good model. It explained 89% of the turnovers just with these 4 variables here. Each of these ellipses is a meadow, and it's essentially a confidence ellipse around all the quadrats in this meadow. The first axis explains the most variation. The second axis explains the next most and goes down. I think in this analysis, there were 4 or 5 axes. I'm only showing you the 2, the most important one. So this is showing how kind of this amalgamated species composition changes along these gradients. And the main gradients are one from meadows with a lot of cover, a lot of plant cover, to ones that had a lot of rock. This gradient was where the vegetation was high, to more bare areas where it was short. This is exactly the same thing, except now it's showing the species that are driving this turnover in composition. And we're just going to pay attention to 2 of them because they're the 2 most dominant species, and they really dominate things. We've got Carex filiformis and Juncus perii. So Carex is out here where there's a lot of cover, and Juncus is out here where it's kind of dry. What is missing at this small 1 square meter scale? Just a tiny bit of audience participation. No snow cover, no climate effects. Wasn't important. So at this small scale, you have these things operating. Now let's bump up. Let's go up to the plot scale. Now instead of ellipses, each of these gray little circles represents one of those 160 plots. And again, you're getting nice separation along these gradients. That gradient from highly vegetated areas to more rocky areas is still the dominant gradient, although it's not as strong, but it's still strong. Height still matters on the second gradient, but now you have a more landscape scale effect coming in with elevation. Where at that small scale, you had Carex way out here. It's still out there, but it's moved in a little bit, where Juncus is still the dominant one in these more rocky areas. But the point here is we've bumped up in scale. We're starting to see this amalgamation, this interaction between things that happen at a small scale and things that are affecting things at a larger scale. Here's the meadow scale. Still a very good model that's explaining all in all, almost 80% of the variation turnover species. But now those small scale effects have almost entirely disappeared. And what is shaping the composition in these meadows and the species that are driving those are now things like the size of the basin that meadow, essentially a watershed area where that meadow occurs, how variable snow cover was over 20 odd years or so, and then the total number of snow-free days. And this god-awful acronym down here is simply an index of how complex the terrain is around it. So this kind of answers the second question: how important is climate's influence on composition? It only matters at large scales. Other things are going on and interacting at smaller, intermediate, and small scales. And the point here is that species composition is being influenced by multiple things in multiple ways at multiple scales. Okay, so those are the plots that we sampled once. Oh yeah, don't forget the third question: is there a spatial trend in turnover in species composition? Very, very, very minor. It only accounted for about 1/10 of the variation that the environmental variables did. And after a few hundred meters or so, it disappeared. What that means is the plots about half a kilometer away from each other can be just as different in species composition as if they were 50 kilometers apart, or they can be as similar. So you have all these things mixed together. Now we'll get to the repeated measure plots, the plots that we measured multiple times. No fancy graphs for this. The question is, to what degree did composition change among years with high and low snowpack? Boom, it didn't. There was no substantial change in the species composition in these meadows, despite these wild fluctuations in snowpack over essentially a 10-year period. What this is indicative of is strong resistance in these meadow communities to change. And that feeds into diversity. Intuitively, you would think if you don't have a lot of change in composition, you're not going to have a lot of change in diversity. And that's actually how things played out. Okay, just a little bit more of one of those mental exercises. Quantifying diversity was a real mess for decades and decades and decades. But over the last, certainly 10, probably 15 years or so, it's really come together. So how many of you have heard of alpha diversity? Okay, so for those of you who have, alpha diversity is measured at kind of a smaller scale. How many of you heard of gamma diversity? Okay, so gamma diversity is essentially the sum of the alpha diversities. It's the total number of species and their abundances at large scale. Then you have beta diversity, which is the ratio between gamma and alpha. And what that tells you is how heterogeneous your communities are. It tells you how much turnover, it's getting at composition, just in a different way, how much turnover there is. This is kind of cool, the system that's been developed, because it's hierarchical. So gamma diversity at a small scale, like a quadrat, if you average that all together, it becomes alpha diversity at a larger scale, say at a plot, or you pull all the plots together for a meadow. And then that gamma diversity at the plots, or alpha diversity at the plot scale becomes, I'm sorry, gamma diversity at the plot scale becomes alpha diversity. Okay, so it's hierarchical. The thing I want you to pay attention to are these curves. Indices have different sensitivity to rare species, and you have to account for that. And that's what this Q value is, and that's all I'm going to say about that. So here at Q equals 0, that's equivalent to species richness. And as you go out, you start incorporating abundances, cover data into it, and you get these curves. And what this allows you to do is, if these curves don't overlap, you can say unambiguously, this is more diverse than all the rest of this. If they cross, then you have to start qualifying it. You can say, well, they're more diverse here, but they're just the same as out here. So it's a really comprehensive way at getting at this stuff. So cutting to the chase, these are the plots that are sampled once, and these are called diversity profiles. Using these profiles, the Booman factor, the thing that really matters at the alpha diversity level, well, it doesn't, alpha, gamma, beta. The thing that really seems to be shaping diversity in large part is parent material. The soils that these meadows exist on that were derived from the parent material. And the one that really has very high diversity are the volcanics. Granite, kind of intermediate. And I think intuitively you know it, granite's not super rich in terms of nutrients and stuff like that. But regardless of scale, when you're looking at alpha diversity, those volcanics are really driving things. And just to kind of give you a feel for it, since we're talking about a 1 square meter scale, and this is equivalent to species richness, here on average, you're picking up about 7 species in a quadrat. Whereas if we pull things together and plot species richness, you get about 35.25 hectare area. So that's how you interpret those. And that's going to get important here when we get to beta diversity, because things are a little bit different with beta diversity. Soil still matters. If you look at this small scale across all these quadrats that we had, just based on species richness, we have the equivalent of 14 communities as if they had no species in common whatsoever. So you've got a lot of turnover at this really small scale. If you go up to the plot scale, now all of a sudden the granites are higher than the volcanics are. But look at the numbers here. Across these 160 plots at the plot scale, they're really similar. You only have the equivalent of 3 communities with no species in common. And again, getting to the important idea for the plots that were sampled, the repeated measure plots, the ones in multiple years. Here's alpha diversity at quadrat plot scales. You could say statistically that 2017, this green line, was more diverse than the other years, but the difference is utterly ecologically meaningless. It's tiny. Same thing at the alpha at the plot scale. Beta diversity, same thing. The years just did not differ in diversity, and that's not surprising because they didn't differ in composition. So if we pull this together for diversity in these meadows, at a spatially parent material is really shaping diversity, really important. I don't know what the mechanism is. I have some guesses up there. Could be, water holding capacity. I'm looking more closely at nutrients, possibly soil depth. It's kind of a distant third. Where you see the turnover in composition in the meadows, it's at these 1 square meter, these really small scales. But you have really low turnover when you go to larger scales. And there's no evidence of changes across time in these periods of high snowpack or low snowpack. So once again, evidence of resistance in the meadow communities. Okay, so now let's look at biomass production and productivity. And we have 2 groups of questions. One relates to time, one relates to space. In terms of time, we want to know what proportions of meadows look like they were increasing, decreasing, or not changing in regards to something, and I'll tell you what that something is. Well, what we measured was an index of production. What proportion of the meadows have what we call a break point, where all of a sudden they're chugging along and then there's a change. It either goes way up or goes way down. Doesn't become anything different, but you see a real change in the amount of biomass that's being produced. And we would also like to know, if we find breakpoints, are those happening at the same time? And then finally, the third question is, what were the rates of change in the ones that were either increasing and decreasing, how they compare to each other? Our questions regarding space was, again, are there any geographic trends, latitude, longitude, elevation in the change, what's changing where? And then the more interesting one is the second one is how segregated these meadows were. So were all the meadows that were increasing in production here, all the ones that were decreasing over here, all the ones that weren't changing over here, or they all kind of mixed together? So lots of satellite data. Also, time for another shout out. Josh Fears, who is now at UC Merced, he was at the Information Center for the Environment at UC Davis, and he and his team did a great service, and they pulled together all these disparate meadow polygons that have been floating around and were entirely unhelpful, and they pulled it into a coherent whole. And so Josh did that for all meadows, not just the high elevation one, he and his team. Really great resource. And since we're interested in what's going on basically above 3,000 meters, I've got 8,180 meadows that I've been looking at over about a 35-40 year period, because the data that we're using from the satellites that indexes production, we've been able to get pretty reasonable data back to January 1985. After 1991, 92, it gets really good and goes through the present. There's two indices, the normalized difference vegetation index and the normalized difference water index. NDWI, the water index, it tracks how moist the vegetation is. I'm actually going to ignore that because I haven't finished analyzing the data, but I've done a lot of analyses on NDVI. So I'm going to concentrate on that. And basically what you do is you take these data and you develop these monthly time series from which you can derive all sorts of indices of production and productivity. Now, this data is coming from outer space, so we want some quality control. So the first thing we did is we ground truth Josh's meadow layer. And those guys kind of hit it out of the ballpark based on 153 meadows that I visited. What that plot of land was around Josh. The 60% disagreement was it was pretty minor. It was basically drier areas embedded in these lower seepy areas. So for all intents and purposes, they got just about everything right. And then make sure that NDVI actually was indexed in biomass. So we took our biomass data from our plots and looked at the correlation. It's really high. Yeah, we're pretty stoked on these data. We've got a lot of good feelings about them. All right, so looking at you can have either these continuous time series or you can have these breakpoints where things are more punctuated. So feel for that. In time series lingo, those are what's known as a local level model, which means that they're kind of fluctuating at an average, not anywhere. But there's not really an increase or a decrease. Then in time series lingo, you have ones that are drifting, which means that they're going up and down, but they're going up and down. That panel on the left is increasing. The one on the right is one that is decreasing. Then you have your class. This one happens to be a linear decrease. You also get linear increases. These are really rare, like well less than 1% of the time. Then you got these. Here's your breakpoints. Around 1998, in this particular meadow, this index, NDVI, it really jumped up in productivity. I don't know exactly why for this particular meadow happened, but this is actually quite common. And what I can do is I can use the statistical properties of these types of patterns, and I can group these meadows together into these classes of increasing, decreasing, or not showing any meaningful change. So the panel on the left shows the number of meadows that had breakpoints. 62% of them had at least one breakpoint. And it's not like they all broke point at the same time. It happened across a whole range of years. What's interesting is that last bar that you see on that right panel, that's 2007. There hasn't been any significant number of meadows that have shown a breakpoint since 2007, over the last 14 years. I don't know why that is. It's really interesting. But what I've been telling people is that the meadows, in terms of their function, in terms of this production and productivity, they're like snowflakes. Everything seems to have its own pattern. Very individualistic. About just under a third of the meadows had a downwards trend. So that means that the remainder had no trend and were actually increasing. If you look at that question of where they were, was there a trend? It's sloppy and it's weak. For the meadows that were decreasing, there's this really, again, sloppy, weak tendency for them to be in the south and the west part of the Sierra. And that sort of makes intuitive sense because it's warmer down there. But again, it's sloppy and weak. Whereas the meadows that were increasing or not changing, they tended to be more in the central part of the range. But I don't put a whole lot of stock in these. By the way, I mislabeled that. That's not 1989 to 2015. That's actually 1985 to 2018. So as I add in more data, the pattern, it's actually getting sloppier and weaker. But what is interesting, oops, back up, don't get ahead of yourself, is where these increasing, decreasing, and no change meadows are in relationship to each other. Basically, after a couple hundred meters, everything is mixed together. There's just, you can be standing here and you can go up to a meadow up there at the Owens River, and it's going to be drastically different as one that, say, was up at Mammoth. They're all mixed in together. One way to look at this is to do what's known as a distance decay analysis. And it's called Tobler's law, and it comes out of geography. And the idea is basically things that are close to each other are more similar than things farther away. So I did these pairwise comparisons between all 8,180 meadows to see how similar they were. So here's your distance in kilometers from each pair of contrasts between these meadows, how far apart they were. And this is an index of similarity. If you're at 1, you're basically identical twins. If at 0, you couldn't be more dissimilar. So look where that intercept is. To start with, even at very, very, very close distances, you don't have a lot of similarity to your neighbor. And then if you go out hundreds of kilometers, that's kind of the way things are going. That in and of itself is interesting. But I find this even more interesting, that huge spread around that line. And we got some reinforcement of this from the plot data, looking at plant height over this period from 2009 to 2017 and biomass. So here's the signals, but here's the noise. Here's the 140 odd species in the meadows, and here's the range in mean responses. Here are the mean responses, the range in mean responses for the plots in terms of biomass. Where's the information? Is it the trend line? Is it the mean? And this brings us to the mammals. Now, I know you guys are plant people. Bear with me here with the critters. And I know you all do a lot of hiking in the high elevation stuff, but don't take these guys for granted. These are really cool species. These are the 4 species that we've been working on, and I'm going to go off just on a little tangent, get up on my soapbox. The pika in the Sierra Nevada is doing just peachy, thank you. It's doing fine. Other parts of its range, it's got some problems in parts of its range, but not all in all. All this hoo-ha about pika has just been, I will stop. I said my piece. So there's the mammals that we're looking at. And here's the fundamental question. What's their effect on establishment of difference? We looked at this in 3 different ways. We wanted to know the input of seeds from the conifers into the meadows, this thing called a functional response is how an animal responds to varying levels of food. So is it eating more of the food when there's less available, more available? And then where we put up hundreds and hundreds and hundreds of these little exclosures, and we put seeds and seedlings into them. And you can see the expression on that marmot's face, the body language kind of says, "Oh, what are you doing? I know there's something really good in there to eat." And these exclosures work great. Had a site up at Evelyn Lake in Yosemite and a site up in the Ducey Basin. Okay, so let's start with the seed rain. I put out these seed traps at varying distances. And this is basically what you found. After 20 meters, you don't get a lot. You do get a very, like 1/100% of the seeds actually. You can see the little ticks out here. 1, 2, 3, 4, 5, 6. 6 seeds made it out to about 45, 50 meters. The vast majority of seeds are coming in within 20 meters or so of the propagule source, patches of conifers. This is the functional response. This was an experiment I did one year, 2011, and I put out 11 different seed densities and then monitored those over a series of, I think it was 10 days, and saw how many were removed, and I would replenish them each day, so on and so forth. And these are exactly the same data. It's just that this is expressed as a proportion. And the reason that you do that, it helps you identify what kind of functional response it is. I'm not going to go into that. What really matters is this right panel, the number of seeds that were removed. Certainly there's density dependence here, but the ones that occur at low density are the ones that are most likely to escape getting eaten. In other words, if you're a seed and you want to get established in a meadow, you're going to do it on your own. You don't bring your family, you don't bring your friends or anything else, because those big furry things are going to gobble you all down. Okay, here's the experiment. At both sites, we had 3 arrays, 84 stations per array, and they were divided up between being seeded or unseeded and within or outside of an exclosure. And I also used different densities, but I don't have to go into that level of detail. We also had a separate group of exclosures that just had seedlings in them, and we planted those seedlings in over 3 years. This was a lot of work, but we got a lot of help, which wasn't necessarily asked for. So this is up at Evelyn Lake in Yosemite. There are 10 biologists, 6 mules, 2 horses, 2 packers, and probably 10 million mosquitoes. And that's my fanny pack, my butt, my back. You see there on the left, covered in mosquitoes. And you can see the crews with their mosquito nets on and stuff like that. I'll tell you, this was great fun. We have all sorts of war stories from establishing this. And the patterns just could not be more stark. The panel on the left is the number of seeds, or the proportion of seeds that germinated inside the exclosure, which is the green line, and outside the exclosure. And it is along a moisture gradient. If you were not in an exclosure, you were going to get munched if you were a seed. And if you were a seedling, it was exactly the same. So what's going on here? Well, the mammals are having a major effect. Now, those are experimental sites, 2 experimental sites, well replicated and all that sciencey stuff. But we can use our plot data and see where conifers are occurring in what plots, how far those plots are from a propagule source, the nearest patch of conifers. So we've got our adult trees, our matures, we've got our saplings, and we've got our seedlings. Here's your distance from the propagule source. This is the density and the belts that we were doing these counts in our plots. This is a classic distance decay function. This is what you expect with distance. But again, even for the mature trees and the saplings, most have been established at pretty low densities within a couple hundred meters of these sources. That is not the case with seedlings. There's no distance decay, extraordinarily low numbers. What this is telling us is that yes, conifers establish in meadows, but at really low densities and at really, really slow rates. Not surprising, things happen slow in the alpine. I call this the gauntlet hypothesis. If you're a seed, you've got to go through an ecological filter. First, you hope you get to a suitable place. Then you hope that one of those big furry things doesn't gobble you down. And if you get that far, you hope that you have arrived in a series of time, at a point in time where you don't dry out. So you've got three things happening here: dispersal limitation, predation limitation, establishment limitation. With the mammals playing a huge role. So this is what we expect these nice defined communities to develop over the next century or so because of simply climate, or something that's a lot more patchwork, kind of like what we have right now. Because there seems to be a lot of resistance in these meadows, a lot of variability, but they're showing resilience. And the mammals are essentially managing the meadows. Now, some cautions. The first thing is thresholds. This keeps me up at night. There's actually not a lot of empirical evidence for tipping points and thresholds. You hear the terms all the time, but there's actually very, very, very few good examples of them actually happening. But the theory is interesting because the theory predicts that the levels may not change all that much as you approach a tipping point, but the variability goes like this. And what have I been blabbing about, going on and on about variability? So at night, I think about, well, what if this is what we're seeing? But it's something that I'm taking very seriously. I'm not sure we've seen the worst yet in terms of temperature or precipitation patterns. I will say that the droughts that we're experiencing, not monsoon. Monsoon has actually been higher than the average over the last 10 years. And then the last point is the alpine is not representative of things going on at lower elevations, even in the subalpine zone. Landscape context is different. So once you get below this kind of magical, arbitrary 3,000 meter number that I'm using just as a benchmark, the meadows are actually embedded within this matrix of conifers. And it's exactly the opposite when you get up into the alpine. So you get a lot more propidial pressure coming into the meadows. I don't care how hungry the mammals are, they can't keep up with that kind of propidial pressure. My last shout out, Caitlin Lebutkin did her PhD at UC Merced, and she was doing a study at the same time I was at. We overlapped a lot. And Caitlin did a really nice job showing that in some of the meadows in the subalpine zone, they do seem to be undergoing this transition. So there are some things far from done with this. I think the most important thing that I need to look at over the next year or two is have competitive interactions been shifting. I've been concentrating a lot on this environmental filtering. So once you there are ways of removing those effects of the environment and then looking at interactions between species to kind of get a feel for competition is increasing or decreasing. But to me, the message is pretty clear. It's real. We have to remove ourselves from averages, means, trend lines, and think more in a variability context. And I don't think that there's going to be uniform responses. And I don't think, I'm positive it's not going to be driven by climate alone. So what's the signal here? Well, that's where we get back to the title of this. I think the noise in a lot of instances is actually going to be what we call noise is actually what we should be paying attention to. In terms of implications, the local effects are going to modify a lot of larger scale effects. I think we have to stop thinking in terms of things like vulnerability. different frameworks, and I think stability is a useful one. And to end with a note of optimism, just because things change doesn't mean it's a disaster. That you stayed awake.
You're happy.
Right?
Have people done similar work in other ranges? Because I'm thinking like the Sierra Nevada is kind of weird with its Mediterranean climate. Mostly perennial alpine species, not a lot of annuals. Like maybe more variation with climate and like the Rocky Mountains where you have that can quickly explode.
Yeah, definitely thought about it. There has been work along these lines in Asia, Scandinavia, a little bit interesting in the higher mountains, tiny bit in New Zealand, and nothing. And this isn't an ego statement. Nothing quite as comprehensive as this. I'm showing here have been documented. So I said the Sierra is kind of weird. You know, the Cook's Range, you use South Island, New Zealand, that kind of wind. Every mountain range is unique. Aerodynamics that we have considered. And so taking the way you frame that question, what I just said, I would say that these mountain ranges on a global scale are the noise that I'm talking about. Consistent response among mountain ranges globally. And you think of what's going on in Asia, and the Andes too, for that matter, those things have been so heavily impacted by grazing. Now, yes, we've had grazing, sheep were run up, you know, from Tahoe to Carson City and all that. And grazing. Nowhere near the levels or the duration. So things like that. So that was a really good question.
How are you measuring variability by blood loss or different types? How do you do the noise, the variability?
If you're looking at temporal variability, you can do it literally on a quadrat by quadrat basis. How's it fluctuating over time? You could do it on a plot basis, how things at the plot level are changing over time, meadows and stuff like that. So basically you're looking at how much something is hitting a maximum to how much it's hitting a minimum, and then everything in between, all of that. The actual measure, there's two measures that I'm using with these, I call them temporal dynamics, which is variability over time. One is the coefficient of variation, which is simply the standard deviation of something, and you divide it by the mean. Another is a little bit more sophisticated. It's called the consecutive disparity index, and it fleshes out the coefficient of variation a little bit because it tells you how consistent, say, down years are relative to up years, or are they all mixed? So I'm using these two different types of measures. Spatially, there's all sorts of different ways of doing it. I did not want to even begin to get into the statistical part of all this stuff. You definitely would have your heads on your shoulders and snoring by then. But you can do things where it's called hierarchical modeling, and you can see if things that are happening at a small scale in one area are playing out the same way in another area. And that's how I get those credible intervals around those zero lines. So
other people.
Yeah. Well, you don't need huge samples to do this. My intention originally was to have maybe 40 or 50 plots, and we ended up with 3 times that many over that. And I think that I wouldn't mind having a few more in just a couple areas that we've missed. I don't want to go back and do that now because they're really out of sync and out of sequence. So yeah, maybe a few more, maybe up to 200 or something like that. But I think we can tell a pretty good story because it captures so much of a range of variability in that high elevation zone. But yeah, the people thing, my God, if I didn't have it at the peak when I had the office here in Bishop, I had 26 people working with me, and we needed every single one of those to get this thing rolling. Yeah.
Could you tell us a little bit more about the soil differences? First of all, I wondered, what area did you actually find volcanic?
So I just realized I have a Dodger game I have to go watch over at Rusty, so I'm getting out of here. The volcanics are basically intrusions scattered throughout the range. There's a few spots. There's one area up towards, I call it the Girl Lakes. It's the northeast corner of Yosemite. There's Ruth Lake, Muriel Lake. How many of you been up in that? Yes, very northeast corner. And there's some nice there's a fairly large area, maybe a few square kilometers of volcanics up in there. Everything else are just these little intrusions. You can tell where they are though in the meadows because the soil tends to be darker, a lot finer. That gets kind of lost in really wet years. There's a lot of soil moisture, but in drier years it becomes fine.
So I think I followed you that the difference in the soils really got lost in all these snowflake meadows and the variability of everything. But did you see?
I think you're okay. No, you go ahead. You finish.
Okay, but I'm wondering if, to the casual eye, would you see different species coming out of these soils? Would you?
The soils were related more to diversity composition. So you saw more species in an area, but they're the same ones that are occurring in the group. You just hit the nail on the head with what you were saying, how all this stuff is intermixed. What we're seeing with the vegetation, I think is simply reflects the heterogeneity that's already there.
Yeah.
Long established.
Yeah, that's right.
Are you anywhere connected to that knowing network? That when they started several years ago, National Ecological Observation Network? Neon, sorry.
Yeah, no, I am not. They're like the LTERs that NSF runs. They're specific sites, and so no, this has been simply an opportunity that came about around 2007, 2008, and we've been able to make it work here. I wouldn't mind being part of a network like that. Administrative headaches that come along. I know somebody, a friend of mine, is deep into the Neon network, and she's an outstanding scientist, but basically she's had to morph into an administrator. So that's just the nature of these kind of things.
Looks like that's it for questions, and I don't see anything on the Zoom chat. So
Okay.
That's it. Thank you very much, Rob.
Thank you. Because the Padres drive me crazy. I was so aggravated last night. My God, so an extra inning game